It's an honor to be invited to give this talk, and what I'm hoping to do for the next 30 or 40 minutes is to look at briefly at the trajectory of the COVID-19 pandemic, to talk about two examples of success in science and a little bit about their accompanying challenge that have happened with the pandemic, and then to think about where we're going with COVID and what the future holds. And I'll apologize ahead of time if you were expecting a basic science talk. I looked at all of the other presentations today and thought there was a huge amount of science. And so this is a little bit more of if you like, a policy and impact of science discussion about COVID. Okay, next thing, disclosures. I've done quite a lot of work with vaccine companies, and we're not talking a lot about vaccines, but just so everybody's aware of both my work with and for vaccine companies and with grants from a number of other both nonprofit and industry funds. All right, so let's start. Here's the very beginning of the pandemic in 2019 and 2020 with a first reported case of what was thought to be coronavirus infection associated with the Hunan wholesale seafood market in early December. The first case reported outside of China on January 13th, and the first case identified in Canada on January the 23rd. So that by the end of January, we knew that this was going to be a global pandemic. We just didn't have a good sense of how quickly it was going to move or how bad it was going to be. So how's Canada done already? This from Our World in Data is a graph of cumulative cases of COVID-19 confirmed, laboratory confirmed per million population. So it's selected countries because obviously the rate of disease that you see depends a lot on lab testing and a number of other variables. So I've tried to select for countries that have relatively similar rates of testing and capabilities. And what you see on this graph is that there's a really wide range in the world of cases per million in different countries. And Canada is this green line that ends in a star. So we're pretty solidly middle of the pack. Many fewer cases than, for instance, Sweden and the United States and France, but many more cases than this group of countries down at the bottom that start with Trinidad and Tobago and Singapore and Japan and Australia and go down to Taiwan and Vietnam. So look at this for a moment and think about what distinguishes those countries that have very few cases of COVID. Why did people get to be successful with COVID? And one of the first things about this is it's not about your GDP, right? It's not about how rich you are or what kind of resources you have, because Sweden and the United States are right at the top of United Nations indices for health and well-being, whereas Vietnam and Taiwan are not nearly as close to the top, nor is Trinidad and Tobago. So an independent panel commissioned by the World Health Organization has just recently weighed in on how well people have done and what we need to do to prevent the next pandemic. And I'm not sure I agree with them that we could have prevented this pandemic. I think there's an element of wisdom of hindsight in that thinking, but there's no question that it will be really helpful for pandemic planning going forward to understand why some countries did well and other countries did not. And the short version of this, the link to the full panel report is below, and it's really interesting reading. The short version of it is that there were a number of factors that contributed to control. The first thing that you may have noticed about the countries that have had successful control is almost all of them are islands. And in this particular pandemic, controlling travel has been very effective. This is different from, for instance, influenza pandemics, because influenza moves really quickly and you can't shut borders fast enough with influenza to have much of an impact on influenza pandemics. And that interfered actually with our management of this pandemic, because we're all used to the rules for influenza, which said don't try to control borders, treat it as a global outbreak. But in this particular pandemic, controlling borders was actually very effective. Just being lucky was good too. So many countries have done well, had late introductions, and there's nothing quite like having watched somebody else live through it to know what you have to do and do it better. But a big piece of this is about having strong, adequately resourced public health systems and about having done a lot of pandemic planning. So Taiwan in particular had a devastating outbreak of SARS-CoV-1 in 2003 and spent a lot of time working on strengthening their public health systems so that they would do better when the pandemic hit, and that has really paid off for them. Singapore and Vietnam are two other countries with really strong, well-resourced public health systems. In contrast, in Canada, our public health system really struggles. So our total spend in public health in Canada is the equivalent of 3 days of what we spend on hospitals every year. Really badly under-resourced public health system. And so our contact tracing, our systems for control of transmission were very rapidly overwhelmed at the beginning of the pandemic in Canada. Interestingly, though, one of the perversities of our control measures is one of the things that's helped us control this pandemic in Canada has been the fact that we have, relatively speaking, very few hospital beds and even fewer ICU beds per capita. So when our cases go up, we hit healthcare system catastrophe much sooner than other countries. And because, of course, governments and citizens respond to health system capacity issues, that has actually contributed to our ability to introduce measures to reduce hospitalization and ICU admission when we need to. And that's, for Canada, been a significant piece of keeping the numbers down. So what does the pandemic look like in Canada? This is the U of T Dalla School of Public Health groups mapping of daily COVID-19 cases in Canada. And the first thing I just want to point out is it looks, when you look at this, as if the waves of COVID are getting larger. So a small one in March, April, May of 2020, followed by a long, slow increase with a peak in January, followed by a third wave that's just past peak now. But in fact, this peak in April and May looks much smaller than the actual impact, because you have to remember that we didn't have testing at nearly the same capacity in April and May. One of the really interesting and challenging things about this pandemic is, again, we've been preparing for influenza pandemics. And when influenza pandemics come, they come in these really distinctive waves that occur at different times in different parts of the world. And so you don't need a global supply chain that can supply everything to everybody at exactly the same time. But that was what we needed for COVID. And so we quite literally ran out of everything. One of the really striking things about those first few months is we didn't have nasopharyngeal swabs. Okay, 30-cent things, they sell for about 90 cents. Much of the supply for the developed world came from a plant in Italy, and Italy was shut down because of their COVID outbreak. And so the entire testing system was held hostage to the fact that we couldn't do testing because we didn't have NP swabs, and never mind a whole lot of other shortages in testing reagents and in setting up testing in microbiology labs. So if you want to get a relative sense of the size of the first two waves, you can look at a recent publication from the Science Table in Ontario. This is an analysis of cremations in Ontario, and the reason for cremations is that cremations are somewhat newer than burials, and so there's an electronic system for cremations in Ontario that's got data that you can access much earlier than you can access the usual CHI stats on cause of death across the country. And so the blue here is the weekly average number of cremations from 2017 to 2019 by time of year. The yellow is the weekly number of cremations that were not counted as COVID deaths in excess of baseline. And the orange is those numbers plus the COVID deaths. So you can see a couple of interesting things here. The first is that there's an excess rate of death that appears not to be directly due to COVID, that we're not sure what it is. So it could be increase in opioid deaths because of people staying home. It could be people not being willing to go to the hospital with cardiac problems, for instance. But at the beginning of the first wave, it was us not making diagnosis and not recognizing disease. And you can actually see that the number of deaths in the first wave is of the same order of magnitude of the number of deaths in the second wave in Ontario. So that that appearance of much less disease is just a testing artifact. So the other thing about this is we've seen in Canada what we've seen in many other countries, which is what we're calling waves of disease. I'm actually still struggling with us calling them waves because to me, a wave of something is something that happens from natural causes and is not controlled by humans. So waves of influenza have nothing to do with how humans behave. They have to do with how the virus behaves. But waves of COVID, of course, are completely controlled by human activity. So you get a wave of COVID when you reduce restrictions, you have more contact between people, you allow the virus to spread. And you end a wave of COVID by instituting public health measures that restrict activity, that stop contact, and that bring the virus back down again. And every country in the world has been involved in this, if you like, backing and forthing, in which we try to relax public health measures so that we have less of an impact on other activity in society. And when we do that, we allow spread to start again. Because of the delay in when you see cases, as opposed to when they're actually transmitted, and because, of course, spread increases in an exponential fashion as opposed to a linear fashion, you get trapped in these very rapid increases and then you have to wait for it to come down on the other side. I'm going to come back to this wave a little bit later. Okay. So now let's talk about a couple of what, in my view, are remarkable successes of science in this pandemic. And the first of them has been our ability to put together really large randomized controlled trials to do rapid assessment of whether we can find new treatment for COVID-19, right? New virus. No idea what works. You can do a bunch of testing in the lab, but testing in the lab doesn't necessarily tell you what the pathophysiology of disease are or what's going to work. And the most remarkable of these is the recovery trial run out of Oxford University in the UK. That is a simple randomized controlled trial. Its outcome is death at 28 days. They got the government and all of the public health departments to support them. And over the last 15 months, they've randomized more than 40,000 patients in hospitals all over the United Kingdom. And they're responsible for the first reporting of most of the positive effects. So dexamethasone, tocilizumab, remdesivir, all of those effects we have primarily because the recovery trial has delivered them. It's a phenomenal achievement. But there have been some other really good things in clinical trials. So the World Health Organization, at the same time as recovery was being developed in the United Kingdom, developed what's known as a solidarity trial. So that's an adaptive clinical trial for different COVID-19 treatments that has, in the last year, randomized more than 12,000 patients, 500 hospitals in 30 countries. CATCO in Canada is the Canadian arm of the solidarity trial, and we've randomized 1,476 patients in 58 hospitals across the country. And while that's, we would all have liked to do much better, that really is a remarkable achievement. We have not had an infectious disease consortia that can set up and run randomized controlled trials. And so our ability to mobilize has been a really significant achievement. Here's another amazing achievement, Concord 1, which is a trial run by hematologists with the collaboration of Canadian Blood Services and many other people across the country, looking at convalescent plasma for the treatment of COVID-19. That trial was stopped for futility not long ago, but in months, that trial randomized 940 patients in 73 hospitals across the country. Again, a really remarkable achievement for speed of getting trials done, particularly in a crisis. Now, there have also been a significant number of challenges with our response to this, and some of them pointed out in a recent Nature commentary. One of the difficulties is that when people did in vitro studies, everybody could see those in vitro studies. And so lots of people wanted to start trials, but because we don't have the capacity to get people to work together and to set up consortia for clinical trials, means a very large number of clinical trials got started by individual groups for all sorts of drugs. So 250 registered trials for hydroxychloroquine. And most of those trials were too small to be useful. And while you can argue that if that's what meta-analysis is for, you put trials together so that you can get bigger outcomes. Truth is that most of those trials are also not really high quality and not that useful. So there has been a substantial number of, a substantial amount of really wasted effort in getting our clinical trials together. And we need some very careful thought about at a global level, how we manage this going forward. And there are some other significant challenges about how we manage clinical trials. In Canada, we were fortunate to have the Canadian Critical Care Clinical Trials Group, which is a consortium across Canada that Rob Fowler and Srin Murthy and Deb Cook and a number of other people have been putting together for a long time, that pivoted to look at infectious disease trials in hospitalized patients. But we all recognize the need to have something better established. And even that trial ran into really substantial problems with the administrative burden associated with doing clinical research in Canada, with data sharing agreements and contract issues, and with an approach to funding. The funding for true funding for CATCO took months to come, and while months is a really short timeline on a normal scale, it doesn't allow what happened in the United Kingdom, where people could just focus on getting the trial up and running as opposed to worrying about how to fund it. In part because we have the capacity, our coordinated approach around the world actually has been focused on platforms for treating inpatients in hospital, but we really need a coordinated testing platform for outpatient studies, and that has not happened. And we also don't have a testing platform that works well for new agents. So there are now a number of new antivirals in phase 1 and phase 2 trials around the world. And those are being obviously developed by pharmaceutical companies. And there are very strict rules about how you do those trials that make that process relatively slow. And while those rules are good, figuring out how we get a platform for pandemics that allow us to meet the requirements for really good clinical trials of new drugs that cope with the outcomes that pharmaceutical companies need from them, but that get us rapid results is a very significant gap that we need to work on. And finally, in Canada, one of the things that slows us down is that we don't have a research mandate in our public health units very strongly. And so public health units are really focused on control and public health measures. And in contrast to the UK, where there was a big move for public health and the research networks to work together to get data out, we have that gap in Canada that we need to fix if we want to do better next time. Okay, second miracle, of course, is vaccines. Just to show you how much of a miracle that is, this is a New York Times article from April 30th, 2020, in which the projected approval for vaccines and distribution was August 2021. And at the time, I think most of us thought that August 2021 was a really optimistic estimate of how much we could shorten the timeline for vaccine development. In fact, two things happened. There's a series of miracles that went into the availability and distribution of vaccines. And the first of them is the amount of work that's gone on in the background for different platforms for vaccines. It is not a coincidence that the vaccines that we have available to us now are new platforms, mRNA vaccines and viral vectored vaccines. And that is because it is easier to do manufacturing and easier to develop and easier to do the manufacturing for those vaccines. So the protein-based vaccines, Novavax, Medicago, GSK vaccine, are all coming later. They may well get approved by August of 2021. That was a realistic estimate for them. But these new platforms that people have been working on that hadn't yet made the grade for any licensed vaccines made it possible for us to have vaccines earlier. The second miracle of vaccines was about Operation Warp Speed in the United States, which was a collaboration between government and industry and academia that tried to take everything possible out of the delays that are usually embedded in the vaccine program. The most obvious example of that to me was the fact that, you know, it was a matter of a week after the vaccines were authorized in Canada when we had vaccines in the country. So what that meant was that we had manufacturing plants that had manufactured vaccines in quantity before we even knew they worked. Okay, under normal circumstances, that's an incredibly stupid thing to do. You waste huge amounts of money if the vaccines don't work, and we did not know that the mRNA vaccines were going to work. But as it turned out, they worked beautifully, and that willingness of the United States to invest in Operation Warp Speed and to make that decision about vaccine manufacturing on spec has made a huge difference to vaccine availability. And the third piece of the miracle is the one I want to talk about for just a moment. So this is a graphic of what the spike protein looks like for coronaviruses. And all of our vaccines are based on the spike protein. one of the major antigens of coronavirus. And it's a trimer. And it exists under normal circumstances in life as a closed prefusion protein. And then when it gets close to epithelial cells, it does yoga. Okay, so the first thing that happens is that the top piece of it opens, and then it undergoes this complex switching where it stretches out so that it can link the host membrane and the viral membrane. And then it folds up to bring the host membrane and the viral membrane close together to allow release of the contents of the virion into the host cell. That's a really complex process, which is actually similar to a number of other viruses. And that's that similarity that's important. So to understand the miracle here, you need to go back to another virus that has a very similar fusion protein called respiratory syncytial virus. Respiratory syncytial virus was first identified in 1956. In chimpanzees, found in humans in 1957. It's a really significant cause of disease in young children and in older adults and immunocompromised adults. And so when the vaccine was discovered, people very rapidly made a vaccine for testing. It was a whole cell formalin inactivated vaccine, which was a common platform for vaccines at the time. And what it induced was enhanced respiratory disease or antibody-dependent enhancement. And there was a catastrophic trial in 1996, 1997, in which the children who got vaccinated when they subsequently were exposed to RSV actually developed more severe disease. And two of the infants in the trial died as toddlers from RSV infection, and that shut down attempts to produce a vaccine to RSV for many years. It turns out that the reason that happened is related to folding of fusion glycoproteins. So this is now a map of or a graphic of the RSV-F glycoprotein, which again is the protein on the surface of RSV that's responsible for fusion with human respiratory epithelial cells. And on the left is the pre-fusion state with one of the best antigenic binding sites on the top in red, and on the right is the post-fusion state. RSV actually loses one of its components making post-fusion and completely loses this antigenic site. When you formalin inactivate RSV, the spike protein fixes in a post-fusion state. And in that post-fusion state, the antibodies that you make to it are non-neutralizing. And when you make non-neutralizing antibodies and then expose the child to the virus, again, two things happen. The first is that those non-neutralizing antibodies then bind to immune system cells, and you get replication and amplification of the virus and release of pro-inflammatory cytokines. And you also get a switch to a Th2 cell response, which gives you release of interleukins without good stimulation of natural killer cells or CTLs. And that results in enhanced disease when you get exposed to the virus and you have non-neutralizing antibodies. So about 20 years ago, maybe 15 years ago, a postdoc came to the NIH in the United States intending to look at actually a similar problem to this with the glycoprotein of HIV. He was talked into working on respiratory syncytial virus. And over time, the group at NIH figured out what the structure of pre-fusion and post-fusion RSV F protein looked like. And then they went systematically on a protein modification search in which they looked at how they could change amino acids in the protein that would fix the F protein in a pre-fusion state that they could then use for vaccines. Took them a long time, but in 2013, they published this structure of prefusion with this antibody binding site. You make antibodies to this site, you create neutralizing antibodies, and they demonstrated protection of infant macaques from RSV infection with this fixed prefusion antibody that had a series of mutations inside the protein so that it could not unfold into the post-fusion state. And it's based on that we now have a series of RSV vaccines in clinical trials for vaccinating infants and moms and older adults that have really substantial promise. So where did they go after that? Well, the postdoc who developed this, now Prof, was looking for the next challenge and started to work on trying to develop a vaccine for MERS using the same approach, knowing that what you would need would be antibodies to the pre-fusion MERS-CoV protein. Turned out that they couldn't fix the prefusion adequately to understand the structure and to try to figure out what they needed to change in order not to have the unfolding into post-fusion. So they went to HKU, which is another seasonal coronavirus, and from that figured out the structure and found the points at which you could change amino acids so that the protein could no longer unfold into the RBD attachment and post-fusion state. And the original version had two prolines inserted into different sections of the amino acid to stabilize it. This new version has 6 of them at these red sides, and it's that modified spike that is present in all of our vaccines. If that work had not been done, if we had not had the work on RSV and the subsequent work on MERS, we would never have been able to understand how to stabilize in prefusion, and we would likely not have had neutralizing antibodies to COVID-19 from these vaccines. So it's just a really amazing story of science in the right place at the right time. And the first really obvious benefit of vaccines is seen in our rollout in long-term care. Canada has had a dreadful time, much worse than other countries, of outbreaks in long-term care facilities and deaths in long-term care. And this is Chris Candel's analysis. So he looked at all of the vaccination in long-term care in December, January, and February, and he normalized each home to the vaccines, the first delivery of vaccine, and looked at what happened to the risk of COVID over the next 30 days. This is Pfizer vaccine, so most of these people got a second dose of vaccine on day 21 or day 22. And you can see that actually even before the second dose of Pfizer vaccine, which is something we know now, you have a dramatic reduction in cases and deaths from COVID. So we've had a 96% reduction in cases and deaths from COVID in long-term care facilities, and stunningly effective mRNA vaccines. Okay, so here we are back to what's going on, and asking the question, what happens from here? And we have two things happening at the same time in Canada. Obviously, we have enhanced public health restrictions and we have the vaccine rollout. And the question is, what's it going to take to, what are we going to see? How is it going to work? And this is really difficult to judge. And the easiest way to do it is to look at the comparisons with other countries that are rolling out vaccine. So on the graph here are the countries that are substantially ahead of us in vaccine distribution, starting with Israel. Israel made a deal with Pfizer to get them data in return for a early vaccine supply for all of their population. And so they are at 63% of their population fully vaccinated. That turns out to be over 90% of their adult population. They vaccinated 19 and overs, and 18 and unders are 33% of the Israeli population, a really young population. So everybody's got 2 doses. They were having a lot of trouble with cases of COVID. So look at how many more cases they've got than we have in Canada until March. But now with 2 doses in, they're down to the equivalent of about 30 cases a day in Ontario. About a third of those cases are from outside the country, and most of the remainder are in the small proportion of people who in communities that they've had trouble with some vaccine hesitancy. And so they have substantially reopened within the country, although they're not still open to outside travelers. So it took, their vaccination program started in January, took about 3 months with not very many public health measures, but a lot of vaccine. The United Kingdom, in contrast, in the fuchsia here, went into a really hard lockdown after Christmas with the spread of the variants. They're now at 54% with first dose, 30% with second dose vaccine, but most of their decline is probably associated with their public health measures as opposed to their vaccine, and they're currently running about 5-fold fewer cases than we have in Canada. The United States, a decline is mostly based on vaccine, and we're at about the same first dose now, but we have many fewer people with second dose. And we're only just a little bit above the United States, probably because our public health measures are stricter than the United States. But the second dose, we need the second dose vaccine to get the numbers down. So this just tells you that it is really hard to judge how quickly we're going to be able to get out of this second wave. Very clearly, we're all tired of the pandemic. There's not nearly as much adherence to public health measures as there used to be. And so it's going to be hard for us to get the numbers down quickly. It's probably going to be a long, slow summer of decline. Yeah, if you want to think about the challenges going forward, we need to talk a little bit about what's been happening to evolution of the virus and variants. We were all really happy when we looked at the rate of SARS-CoV-2 mutation, which is slower than many other viruses, including influenza and SARS-CoV-1. And then right at the very beginning of the pandemic, there was a new mutation induced in spike, D614G, which resulted in enhanced transmission, maybe enhanced virulence. And within a matter of a couple of months, the D614G mutation took over globally, and everything is D614G now. And when it first happened, all of us were worried that it was just the beginning. But then for many months, nothing very much happened. Not that new lineages didn't appear, not that the virus didn't change over time, but there appeared to be no selection for particular viruses. And you can see here an analysis from the group in Kingston looking at Ontario data, in which there's expansion of different clades, but really nothing that takes over, nothing that increases significantly over time. And we all got a little casual, honestly, about what was going to happen with the virus. And then in December 2000, and I'm sorry, the animation's not working on this slide. December, just have to look at it. December 2014, England reported a new variant called B.1.1.7. And you can see here the spread of that. So it started in this southeastern corner of England, first recognized in September of 2020. And you can see its very rapid spread across England. And this is a relatively incomprehensible graph in which they're trying to put 16 things at once. But what you see is the bigger squares and triangles and diamonds are areas where there's a higher proportion of B.1.1.7. And all of them are, and here's the reproductive ratio of the variants. And what you can see is where you have few variants, the reproductive ratio of COVID in December of 2020 was about 1.2. It's not great, it's still expanding, but the reproductive ratio of places that had high proportions of the variant was nearly 2. That doesn't seem like a huge difference, but let me show you what that means in Ontario. In Ontario, we had no B.1.1.7 when we did a point prevalence survey at the end of November. We had nothing when we looked in the 3rd week of December, but variants started to appear in the beginning of January. And by March, you can see that they're making up about 40% of all of the COVID. Now that doesn't look like a very big deal, and the cases aren't going up very quickly. So let me show it to you another way, okay? What was actually happening during this time was that we were maintaining a reproductive ratio of just under 1 for Wuhan and the early variants on the right-hand side in the blue. But B.1.1.7, there were many fewer cases because it started at 0 at the end of December. But the increase is really dramatic because the reproductive ratio is significantly above 1. It doesn't have to be dramatically above 1 because of that exponential increase from a set reproductive ratio. And so everything that happened in the beginning of the 3rd wave, this whole curve, beginning of the curve, is actually not about us releasing restrictions. We were maintaining restrictions. It's about B.1.1.7 moving in and taking over and being more transmissible than the old viruses. So it's a pandemic within a pandemic. And what got us into trouble was not recognizing soon enough that we needed to enhance measures to deal with the introduction of B.1.1.7. The second problem with B.1.1.7 is it's also more severe. Okay, so if you get infected, you're more likely to be hospitalized, significantly more likely to be admitted to the ICU, you're significantly more likely to die. And so it's really been a struggle over the past couple of months in hospitals and in the community to deal with this new variant in Ontario. And it's not, of course, the only variant of concern. We're now up to 4 what are called variants of concern. So these are variants that are significantly more transmissible or significantly more severe or have the potential to escape vaccine, and all of them have at least 2 of these characteristics. If you look at the mutations in them, what you'll see is that these are lineages, they're not single mutations. So most of the mutations occur in spike, not surprisingly, but they have mutations through the genome, and some of these mutations are similar. So 1.1.7 and 351 and P1 all have a mutation N501Y, and that's not because they're derivatives of the same thing. This is convergent evolution of viruses to viruses that are more successful than other viruses. We know that B.1.1.7, because we have data from the UK, has a very significant increase in transmissibility and its virulence has increased. We don't know how well B351 and P1 compete With B.1.1.7, it's been variable in different countries, and we really don't understand why. And B.617.2, which is the variant from India, where is a variant of concern because in the UK, in some cities, it's been competing successfully with B.1.1.7. So increasing in proportion relative to B.1.1.7, which tells you that it is as or more transmissible than the UK variant, which is to worry about. And this is just a map of the proteins. You can see this is for P1, and you can see the concentration of mutations in the spike gene, but the presence of common mutations elsewhere. And what we know about these mutations is variants is, first of all, that B.1.1.7 is not that antigenically different from old viruses, and there's probably a very slight decrease in protection from vaccines with this variant, but it's not significant and will be fine. 351P1 and B617 all have in vitro significant reductions in neutralizing antibody against these variants, and so it's likely that there's going to be a reduction in protection. We have the most data for B351, which is the South African variant. Again, not bad protection, might be enough to get by with the exception of this trial, but clearly reduced relative to the old Wuhan strain in B.1.1.7. So how much trouble we'll get into if these new variants take over is open season. In Canada, neither P1 nor B351 are expanding in Ontario, which is good news. But P1 is about 40% of cases in British Columbia and expanding. So we do need to worry about whether we'll have to deal in the next few months with some reduction in vaccine efficacy. So lastly then, what happens now? You know, this has been a long roller coaster ride in which, you know, good things like vaccine happen and then bad things like B.1.1.7 happens. And a certain amount of anxiety in thinking about the future, because I don't think any of us have adequately predicted the bad things that this virus is doing. So I just want to talk about what I think are the ends of the spectrum of what might happen. So here's our best case scenario. The best case scenario is premised on the fact that the evolution of variants has been adaptation to humans. So this is presumptively a virus of zoonotic origin. When it first started in the pandemic, it was a new virus in humans, and it has been working since then to adapt itself better to us. And so when it's fully adapted to humans, then there's not going to be continuing selective pressure to change, and evolution will slow down and we won't get new variants. And if that's combined with long-lasting immunity from either infection or current vaccines, people are, you know, what you read now is, you know, people who've been infected are protected for 6 months or 9 months post-infection. But that doesn't mean they're protected only for 6 or 9 months. That just means that's how long we have data for, okay? So it could be that once you've been infected, you're protected for a decade or 2 decades. And if infection and vaccines induce long-lasting immunity, and if there's significant cross-protection against variants and new variants, then you know, 2 doses of vaccine will last you potentially a lifetime, and that'll be good. And then that's a last nice thing that might happen would be that of the investigational antivirals that we're working on, 1 or more of them will work, and that will give you something to take if you develop COVID-19 that will protect you from hospitalization and death, and minimize that impact. So if this happens, then the fall and winter of 2021-22 are probably going to have challenges associated with them. We'll go back inside, there'll be more transmission. We're going to really be worried about travel because it's very clear that new variants are introduced around the world by travel. And if you stop travel, you won't get them. And so that's going to be hard for governments to navigate. We're going to continue to have disease and outbreaks associated with unvaccinated groups. We've already got some B351 and P1 outbreaks in long-term care that have been associated with deaths. So long-term care will continue to be an issue. And because we have travel introductions and people want to travel, even if you're vaccinated, of course, you're not completely protected. You go to an area that has relatively high COVID incidence, you're at risk of getting disease. So there will be a series of complications associated with the fall and winter, but post fall and winter, as long as we do a good job of delivering vaccines globally, which is another challenge, then once we get through this winter, then things will settle back down, and by next spring, we'll probably be close to back to normal around the globe. The worst case scenario, I think, is that this is not human adaptation, but just SARS-CoV-2 evolving as it chooses to, and it's going to continue to evolve and to escape new vaccines. If global vaccination programs are limited by hesitancy and adverse events, and that didn't happen in Israel, but it's a significant issue in some states in the United States. If immunity from prior infection or vaccine is not long lasting, if it's only a year or 2 years, then we'll obviously need boosters if we don't get effective antivirals. So if all or most of those things happen, then it doesn't mean that things are not going to settle down to some reasonable approximation of normal life eventually, but it does mean that we're going to have ongoing significant burden of disease probably for some years, and that will be travel restrictions and persistent outbreaks and ongoing disease in the population that will continue to challenge us. And it may mean that we end up needing an annual vaccination program or a vaccination program every 2 or 3 years. Fortunately, okay, we have annual vaccination programs for influenza. We vaccinate a lot of people around the world for influenza every year, so it's not inconceivable that we could do that. And there are already several companies. There's one trial up on clinicaltrials.gov already, and there are definitely others coming, in which companies are making a combined COVID-19 and influenza vaccines, so that if we need to go the route of recurrent vaccination, we will be able to. In sum, I guess with a slight hesitation that we don't know when the next pandemic is coming, and we don't know perhaps what nasty surprises this virus might still have in its stockpile. The worst is over. It's still going to be a challenging next few months getting the case counts down in Ontario, and it's likely to be not an easy ride over the next winter. But I think it's reasonable to hope that once we get through next winter, that as a global community, we will be back to some version of life as we knew it before and through the pandemic. And hopefully we will be thinking about all of the places that we could have done better in worrying about this pandemic. And when the next pandemic comes, we will have improved a number of those things so that our response is more effective. And I want to thank you there. I hope I've left a couple of minutes for questions.
Thank you so much, Dr. McGeer. That was a phenomenal presentation. I truly learned a lot about more of the epidemiological sides of the pandemic as it was progressing. We do have time for a few questions, so if anybody does have any questions, please continue to submit to the chat. I'll start with the one that we have here, and it's by Ariel. So, are the public health measures effective in controlling influenza case numbers and hospitalizations?
Excellent excellent question. So the answer is apparently yes. Okay, I don't think any of us were expecting it. You know, our modeling certainly of new pandemics of influenza suggests that public health measures, while they may mitigate, and there's evidence from the 1918 pandemic that public health measures mitigated influenza, but it didn't stop it. And so none of us were really expecting to see what we have seen this year, which has been an almost complete absence of influenza. This is not just like a quiet influenza year. This has been a non-existent influenza year. To the extent that I think a number of us are wondering about viral interference as well as public health measures. I don't understand viral interference. It doesn't make any sense to me, but it's absolutely true that, you know, when we have a pandemic of influenza, we replace one of the old influenza strains with the new pandemic strain of influenza. Nobody understands how that happens, but it's clearly some form of viral interference in human infections. And so there may be an element of that going on, but it's probably more likely that it's just public health measures. And as we release public health measures, by the grace of God, going into this fall, then influenza is going to come back and just do what influenza usually does. And that will make next season worse, because next season we'll have COVID and influenza, and that'll introduce a new set of challenges for sure.
Thank you very much. Okay, and we have time for one more question. And this is from Eleftherios. So Canada was a global leader in vaccines with Cannot / Pasteur. What happened?
A really interesting question. I think we failed to maintain substantially our research enterprise. You know, we do not spend as much on scientific research as many other G7 or G20 countries, and that's clearly a piece of it. We've also not focused on vaccine research, and we invested very heavily in making sure that we had influenza vaccine manufacturing capacity, which is important. As you've seen in this pandemic, you know, I think many of us were hoping before this pandemic that we had gotten past vaccine nationalism, but very clearly we have not. Okay, and so we know actually that what happened in previous influenza pandemics, and the reason that we have guaranteed vaccine supply, so we have guaranteed influenza vaccine manufacturing supply for pandemics. It's part of our contract with our influenza vaccine manufacturers. But what we didn't think about was that we were going to need some vaccine to something completely different. And we do not have the manufacturing capacity. It's also true that we, in some senses, chose not to be involved in it. We probably could have pitched into Operation Warp Speed. Okay, we couldn't have set up Operation Warp Speed ourselves. That's like $12 billion, probably out of our capacity. But we could have been part of it had we chosen to. And that's another thing I think we need to think about for the next time around is what do we want in vaccine manufacturing capacity? How do we want to be a global player in the development of vaccines for the next pandemic, whatever it is?
Thank you very much. And that's all the time we have for today's session. I'd like to thank you again, Dr. McGeer, for this phenomenal keynote session. And I'd like to thank the audience for attending as well.
Thanks very much, everybody.